Okay, alright. So, hello dears. Ayan, welcome to part 2 of our video. Alright, on sputum culture. And murag ang previous video mention na cut off siya no sa end because murag ni ano na akong memory. Sorry naman. Ng mga iPhone 6 problems. Char. Anyway, alright, 16 GB lang. Char. Anyway, so again, ang point ra ato is pwede siyang i-refrigerate if delayed at 4 degrees Celsius for 24 hours. Okay, alright. Now, for the second part of the video, we'll now focus more on the different types of sputum, all right, or different ways of collecting your specimen, all right? And we'll start first with, of course, expectorated sputum. So, kanina ang sputum, nag-i-expect na nimo, char, na magsige-expect dira, ayaw lagi kay masakitan ra mo, charot lang. All right, so expectorated sputum, by the name itself, it's kusa, kusa na ang patient na nidura, all right? So, it's a primary means of determining bacterial pneumonia. So, primary means lang, guys, but it's not saying that it's the best, okay? So, what it means lang is, it's the most common way para makuha og sputum. And that's expectorated. Kanang muluwa ka. Alright? That's expectorated sputum. It's suitable for adults and older children. Why? Because again, they know how to make dura. Or duraan mo. Ay? Yes. Alright. Yeah, they know man. They know how to expectorate or to dura or to, sputum. Kaya mga children, mga babies, dili, of course, sila kabaluan. Alam mo na, uy, pag dura, di siya kabalo, di ba? So, ato na siyang, suitable siya for adults and older children. Again, dapat walay food and dapat na ang mouth should be rinsed, okay? Bisag with tap water lang. Because again, we don't want na mga food morsels, maapil, like mga like na mga corned beef na or like mga rice grains na maapil. So, klaro kayo na murag, sana all nakapamahaw. Charot lang. Okay, so again, dapat walay food na ma-ingest 1 to 2 hours before collection. And dapat mag-gargle again, no? I-gargle good. Dili nila siya i-spit or swallow, char. Dapat gargle with saline or pwede na tap water. Just again, para mawala itong mga, sa mouth, okay? Para mawala po ng contamination, at least ma-minimize, okay? Alright, and again, dapat deep cough, and dapat ang specimen is duol na sa baba sa patient. Of course, para naman makuha niya ang sputum, alright? Okay, and again, disadvantage contaminated with the upper respiratory tract secretions and normal flora. Okay, alright. Next, we have induced sputum. So when you say induced, imo siyang trigger, alright? Or imo siyang painom or gipasimot og something na naka-trigger niya na mo produce og sputum. Okay, so dili siya mismo ang nag-expectorate ana. Okay, so you induce it. So imo siyang gipagimok or imo siyang gipagawas by a means of an external help, something like that. Okay, all right. So yeah, if the patient is unable to expectorate, like di niya kaya, example na like ICU ba siya or unconscious, etc. So that's what you do. Induced sputum. Again, stimulate production. Aerosol induction. So what you do is you let the patient parang breathe an aerosol solution or something. Tapos that aerosol solution will parang trigger or murag mukatuli ahang diri sa lungs para mo initiate og reflex action or action to expectorate the sputum. Okay? All right. Or pwede pong postural drainage for respiratory therapy technicians or thoracic percussion. All right. We'll look into that later. Ayan, so aerosol-induced, postural drainage, or thoracic percussion. So, gihimong drum, charot, ang patient. Okay, all right. So, we have aerosol-induced. Again, by the name itself, you let the patient inhale something, an aerosol solution. And it's usually used for mycobacterial or fungal disease, no, for isolating. Again, high diagnostic yield for pneumocystis urivetsii pneumonia. Now, pneumocystis urivetsii, it's a yeast, okay? It's a yeast. It was thought to be a parasite noon, but it's actually a fungus. Okay, it's a fungal infection. And this is the most common cause, all right, of pneumonia, all right, in patients with, in HIV patients, all right? Okay, or kanang padulong na to AIDS, okay? Or in AIDS patient, okay? In AIDS patient, the most common cause of pneumonia is pneumocystis urivetsii na pneumonia. Alright? Okay. So, high diagnostic yield ang aerosol-induced sputum for pneumocystis urivetsii. Again, ayan, patient is allowed to breathe in aerosolized droplets of 15% NaCl and 10% glycerine. Alright. And for roughly 10 minutes, and this solution or this aerosol solution na gi-inhale sa patient it triggers again a strong cough reflex. And in that same way, makaproduce na noon siya og sputum. Alright. And secretions obtained are usually watery, but they contain material directly from the alveolar spaces. So, diretso gyud diri sa imuhang alveolar spaces. So, it's really representative of the lower, or like makitaan gyud mo tong sa lower respiratory tract. And it's important for pneumocystis cerevisiae because again, your fungus or mga fungus cells dito sa pneumocystis cerevisiae kay nasa alveoli spaces nagtago or nag-cause of infection. Alright? Okay, although watery siya, but you're expected na naa dito ang mga material from alveolar spaces. Okay? Alright. Ayan. No need for pre-screening specimen. Because again, gikan man gyud siya sa diri, sa imuhang alveolar space. So, you would expect na lesser ang contamination from epithelial cells kaayo or other contaminants. Munang dira kailangan og pre-screening specimen. Okay. Alright. Next, you have postural drainage. Ayan, so you make the patient parang position himself na either maginana or magpatihaya para it's easier daw for mucus to drain so that it may be coughed out. So, samot na if kanadaging gahi kayo na mucus or like tapot kayo na mucus or like plema, alright, so imo siyang ipahigdaw mga inani para mas easy na daw mo gawas or mas mulihok or matangtang ang mucus. Alright? Easy na lang iluwa by the patient. Alright. And again, you use gravity. So, ang gravity will do its job to help drain the mucus into the mouth, again, by placing the body into specific position. So, postural drainage. So, you allow gravity to let the sputum out, okay, para murag mo, easy lang siya move, okay? Alright? Para mas ma-expectorate siya sa patient. Okay. Alright. Next, you have what we call thoracic percussion or chest physiotherapy. So, you use your cup-shaped hand and then imo daw parang percussion or imong ipaigo sa patient para murag ang vibration daw will trigger the airways on the lungs para ma-move ang mucus. Alright. From the smaller airways into larger ones which can be coughed, where it can be coughed up. Okay. So, imong gamiton ang vibration sa inyong kamot. So, percussion. Himon lagi mo siyang drum ang imong patient. Alright. Para mas mulihok ang mucus. Alright. The vibrations move the mucus into larger airways para mas easy na lang i-cough up by the patient. Alright? So these are all induced sputum, meaning kailangan pa external something para mapagawas ni patient ang sputum. Okay? Alright. Ayan. Next, we have your gastric aspirate. Now, for gastric aspirate, by the name itself, it's an aspirate gikan sa stomach. Now, asa siya gikan, ginagamit, it's usually for acid-fast bacilli isolation. And ginagamit siya for infants or young children because ang rationale ani is, habang natutulog daw si baby, alright, so if mo-cough siya, alright, or if naay, like na siya sputum, or mo-cough gani siya na naay acid-fast bacilli, dili niya maluwa, iyaharang ilunok balik, so iyang iswallow. And this na sputum na naswallow is of course, makita na sa stomach, alright? So muna na ay gastric aspirate, okay. You'll, ma-receive niyo muna sa routine lab, actually, naka-receive ko ani sa una. And usually, ang request is for acid-fast staining. Okay. Alright. So, nasogastric tube and withdraw its contents. Oh, yeah. Because again, ang assumption, the AFB were swallowed during the night na natulog si baby. Alright? Kaya kanang giubo daw siya, pero wala niya giluwa because of course, baby, di siya kabalo. So, iharang lunokon balik. Okay? So, muna siya, pag aspirate mo sa gastric contents, mag-expect na kananaa dito ang AFB. Alright? So, muna siya i-point sa gastric aspirate. Okay. Again, must be submitted to lab immediately to neutralize acidity and allow the mycobacteria to be viable or neutralized first and then transported because again, may acidity. So, pwedeng mas okay na ma-transport siya immediately para ma-process para dili kayo mga mawala or there would be a great loss of organisms, especially that mycobacteria imuhang giapas. Alright? Okay. Alright. Next, you have your endotracheal or tracheostomy suction. So, you use Lucan strap. So, it's a really invasive procedure. Kay diretso na sa trachea. So, ablihan diri and then panslakan o kaning Lucan strap. Okay? Alright. So, patients with tracheostomies, they are unable to produce sputum na kanang expectorate. So, you use a Lucan strap to aspirate or suction specimen from tracheostomies. Okay? And again, it should be treated as sputum by the laboratory because again, gikan siya sa trachea. So you have to check for quality of the specimen, lantawon nimo kung daghan bang PMNs, epithelial cells, etc., etc. We'll go to that later. Okay. All right. Now for bronchoscopy, all right. So by the name itself, you use a bronchoscope, all right. So which is a machine na naay murag camera, all right? And then moto ka sa bronchi, okay, sa bronchi, all right. So perform in HIV and immunocompromised patients to diagnose pneumonia. Fiber optic, it's now used to evaluate and manage pneumonia because again, ano siya camera, so makita na. And with this method, the bronchial mucosa, again by the name, as I mentioned, ma-visualize siya directly and collected, makakollect kag sample dayon. And bleeding is a significant complication because again, invasive procedure, magpasulod ka og kani, so could be na irritation na mahitabo and bleeding can also occur. All right. Ang bronchoscopy samples could be bronchial washings or again mentioned bronchoalveolar lavage, protected bronchial brush samples, and specimens for transbronchial biopsy. So again, as you can see, these are all invasive procedures na good because you're really collecting samples again from the inside, from the bronchi. Okay, so you're introducing an instrument already to get samples. All right. Okay. So for the first bronchoscopy specimen is you have the bronchial washings or aspirates. Ayan. So pasudlan siya and then na camera para magay ng doctor. And by the name itself, washing man or aspirates. So naay murag NSS ba to? Saline into the bronchial tree. So imong flushan og NSS ang bronchial tree. And then ang fluid na makuha ato is i-aspirate balik. And mo to siya i-examine. Okay? When purulent secretions are not visualized. Yes. So bronchial tree no naay murag gisulod. Flushan nimog NSS. Okay? And then katong mga fluid or NSS na nakuha or katong fluid na natangtang, all right, or na accumulate from the flushing, mo to siya i-aspirate balik and then mo to siya i-examine. Okay? So bronchial washing or aspirates. Next is of course the bronchoalveolar lavage or BAL. Again, the best specimen jud for microbiology studies in the lower respiratory tract. Again, board exam question daw sa una, not in our time sa una. BAL, bronchoalveolar lavage, all right? So still the same, high volume of saline, 100 to 300 ml, is infused, example, into this bronchi, all right, to obtain the cells and protein. And then after a while, i-aspirate na pod balik. So in a way, same siya sa imuhang bronchial washing, pero mas intense ang volume sa saline diri. All right, and mas daghan imuhang makuha ng mga cells and mga purulent substances. All right, okay, ayan. Okay, next one, we have protected bronchial brush sample. So as you can see, na ay brush na nisulod sa imuhang bronchi, all right? Still the same with the use of bronchoscope, all right? It's the best suited for microbiologic studies, especially for aspiration pneumonia. Now, ang protected bronchial brush manggod kay brush. So basically makuha kag mga cells na gyud. Na example ka na mga nagtapot na sa bronchi, all right? So makuha gyud mo siyang test tube brush na in a way makuha siyang sample from the cells surrounding the tissues, di ba? So it collects from 0.001 to 0.01 ml of material. So it's really kanang grabe ang makuha ba? Sensitive kay ang makuha na specimen. Kay as low as 0.001 makuha niya. So, according to best suited for microbiologic studies, siguro, I think muna siya ang best suited yun. Pero katong question sa boards, murag walay BAL, murag wala ni siya na choice. BAL itong naa dito. So, BAL yun ang specimen of choice. Alright? So, it's either BAL or kaning protected bronchial brush samples. Okay? Alright. Basta, ang point lang yun is, the sputum is not recommended na yun. But it's still used because, again, it's easily collected and dili po kayo siya na naka-invasive. Alright? So, yeah. Okay. And next, you have what we call your transbronchial biopsy. So, ang machine na gisulod na raw is para makakuha na gyud og tissue na gyud na murag mo-cut na gyud og tissue ba sa bronchi. And it's usually used for lung cancer and other lung diseases. So, katong mga na tumors, all right? So, para makakuha sila og sample from the tumor, murag na ay clamp, right? Na murag mo cut from the tumor as a specimen from the tumor, okay? Para again for biopsy. Okay, so again, usually used for lung cancer and other lung diseases. Okay, all right. Okay, next one, we have your transtracheal aspirate. So, muna siya ang transtracheal aspirate. You use a small catheter na insert padulong sa imong trachea by using a needle. So, murag na ay needle na musulod dire, all right? Na murag syringe, and then sa needle itself kay mosulod og catheter, all right, padulong sa trachea to get the aspirate. Okay, all right, ayan. So reduces the likelihood that a specimen again will be contaminated by the URT and diluted by added fluid. So again, na murag needle na ipansok diri or diri sa tracheal dapit, Alright? And then sa needle, pagpasok sa needle, sudlan pod og catheter. Alright? Okay, so inana. Alright? Tapos, ang catheter na ang mukuha sa fluid or sa specimen manggani. Okay? Alright. Rarely used anymore. It's used for isolating anaerobes and agents associated with aspiration pneumonia. Anaerobes, because again, your collection is very closed system, wala na exposed sa oxygen. So therefore, it's really appropriate for anaerobes. Okay? Alright. Ayan. And next, of course, pleural fluid. For pleural fluid aspirate, the term is thoracentesis. Okay, diba sa inyong CM, inyong AUBF, nani siya? Pleural fluid examination. So, mukuha og fluid sa diri sa lungs, okay? And examine ang fluid, all right? Between the space sa lungs and chest wall, mukuha og fluid, all right? And that's thoracentesis. Thora because thorax, diba? Thorax means chest, all right? And centesis is aspiration. So, thoracentesis. So, mukuha og fluid from the chest, all right? Okay. Because of pleural effusion, all right? Again, indicated when pleural empyema. So if nanay liquid or fluid na ni accumulate, there is a space between the chest and the lungs, all right? And it usually reflects the bacteriology of associated pneumonia. Recall that empyema develops if, again, naanay infection daan ang lungs, right? And then wala na agapan, so nagpa-renal infection, so na-accumulate og fluid, all right? So, and the fluid can be purulent, so na mga pus, narito ang mga bacteria na nag-cause na infection, etc., etc. So in a way, the pleural fluid na makuha diri will now be indicative kung unsa tong pathogen na nag-cause sa disease. Okay? Alright. Ayan. And if no material is withdrawn into the syringe after the first try, 3 ml of sterile saline can be injected and then withdrawn into the syringe. So, because maybe wala pa kayo fluid or gamay, so injectan sa nimog 3 ml of saline and then aspirate na para nanay madala, okay, na specimen. Alright, that's for pleural fluid aspirate. Okay. And the most frequently used in children than in adults. Because again, for children, dili kay na nakalisod kay bata pa man. And then for adults pod, less ra pod. Okay, alright. So as you can see here, nanay fluid na accumulate sa lungs. Okay, alright. Ayan. Okay, and next one, we have open lung biopsy. So by the name itself, the most invasive, kay ablihan na gyud mismo ang imong chest, and then mukha na gyud sample from the lungs. Okay? Alright. And surgeons na siya mo perform ana. And again, ang point is, kuhaan na gyud tissue ang lung. So a wedge of lung tissue na gyud. So it's really invasive, okay? Usually used for diagnosing viral infections, pneumocystis pa rin, and mga hard to diagnose or life-threatening pneumonia. If kanang di na gyud siya madala og bronchial aspirates, mga inana. So you go now for open lung biopsy. So kuhaan na gyud nimo og tissue ang lung mismo. Like ang lung na gyud mismo, kata nimo og tissue dira. And surgeon na ang mo perform ana. Okay? Alright. Ayan. So it's really invasive as in. Okay. Alright. Okay. And alright, so now, since we have discussed the different specimen collection, we'll now go to the different, once maabot na siya sa lab, so unsa itong dapat buhaton. Alright? So once maabot na niya itong sputum na specimen, excuse me, of course, first atong buhaton is direct visual examination. And how do we perform that? We use gram stain. Okay. Again, it's used to recognize bacteria and yeasts in the sample. Also used to evaluate, again, the quality of your expectorated sputum by using different grading or classification systems. Diba, recall in your inyuhang sa atong AFB na discussion na ito'y different katong greater than 25 PMNs, less than 10 squamous epithelial cells in ana. So that's the grading na itong i-follow karon po. Alright? So we have different gradings. You have Murray, Washington, Heineman's, and Bartlett's. So we'll start first with the Murray, Washington scheme. For Murray, Washington scheme, ang buhaton lang nato is lantawon ang number of epithelial cells per LPF. If it's less than 10, we accept. And if it's greater than 10, we reject. Okay, alright. So inara na ka siya ka-simple. Ang Murray-Washington scheme. So average number. So kada, let's say, 10 ka LPF, lantawon nimo ang average. If it's greater than 10 ba or less than 10? If greater than 10, reject. And if it's less than 10, accept the specimen. Okay, alright. We also have what we call the Bartlett scheme. The Bartlett scheme, we use, ato siya examine under LPO, low power field, and then na yung mga scoring. Alright, so example, if greater than 25 PMNs, plus 2 atong score. If 10 to 25, plus 1. And if na mucus, plus 1. Greater than 25 sex, negative 2, and 10 to 25 is greater than a negative 1. And what we do is ato siyang i-add, alright? And if ang net score, so example, nakakita kag 25 PMNs, alright? So negative 2. Then nakakita kag, let's say, 11 ka, plus 2 di. If nakakita kag 11 ka squamous epithelial cells, so that's negative 1. So ato na siyang i-add, ang answer is plus 1, alright? So kung ang net answer kani, ang net answer nimo is positive, we accept. Pero if ang answer nimo is 0 or negative, you reject. Okay, so that's the Bartlett scheme for examination. Alright, okay. Alright. Now for the next, or for the last video na gyud nato, we'll now go to the different next criteria, which is the Ayer's criteria. Another criteria used for culturing sputum. And actually, I think mo na itong gina-follow po. Alright, so that's what we're going to discuss on the next lecture until the end now of sputum culture. Okay, alright.